next type of transport is vesicular transport. For the other transport mechanisms discussed, we have only talked about transporting one or a handful of things at a time. With vesicle transport, we are talking about bulk movement of materials. Think about packing things up into a shopping bag and carrying them over the cell membrane. In vesicular transport, things are moving in and out of the cell in vesicles. Vesicles are small membrane enclosed sacs. All vesicular transport requires energy in the form of ATP. The first type of vesicular transport is endocytosis. In endocytosis, things are brought into the cells. There are 3 types of endocytosis. The first type is phagocytosis or cell eating. Phagocytosis is performed by specialized cells in the body called macrophages. Macrophages are like professional eaters. They are very good at eating up bacteria and cellular debris and other things that we don't want in our bodies. When a macrophage phagocytoses something, they create what is called a phagosome. The phagosome will fuse with a lysosome, and the lysosome will then degrade the items. Pinocytosis, on the other hand, is cell drinking. In this case, the cell will form pinosomes filled with extracellular fluids. Pinosomes are primarily full of liquid material. Not a very specific process, it just happens on the surface of a cell. Pinosomes will eventually deliver their contents to the lysosome to be digested. The last type of transport is called receptor-mediated endocytosis. Receptor-mediated endocytosis is a specific form of cell eating. Receptor-mediated endocytosis requires receptors on the surface of the cell that will bind to specific solutes. The receptor bound to their solute will start to form a vesicle. Once this vesicle gets to be a certain size, it will pinch off in the cytosol and is now a coated vesicle. The vesicle will fuse to a lysosome, and once it does, the lysosomal enzymes will remove all the substances bound to those receptors, and the empty vesicles can then be carried back to the plasma membrane for reuse, so this process can occur repeatedly. Exocytosis means solutes will be exiting the cell. In this case, vesicles are formed from within the cell and will fuse to the membrane and discharge their contents to the outside of the cell. Now we're going to shift our focus to the cytoplasm. The cytoplasm is the material between the plasma membrane and the nuclear envelope in the cell. The cytoplasm is divided into 2 components. The cytosol, otherwise known as the intracellular fluid, is fluid having nutrients, proteins, ions, and wastes, and the organelles. The organelles are what perform cellular functions. There are 2 classifications of organelles. There are non-membranous and membranous organelles. The key difference is a membrane, where the membranous organelles are surrounded by a membrane. The membrane is a phospholipid bilayer like the plasma membrane, whereas non-membranous organelles are not covered by a membrane. The first group of non-membranous organelles consists of structures that are part of the cytoskeleton. The cytoskeleton consists of 3 components. The first are the microfilaments. Microfilaments are the solid, slender rods about 6 nanometers in diameter and composed primarily of proteins called actin. The microfilaments are the smallest filaments. Microfilaments have several functions. They can help anchor the cytoskeleton to the plasma membrane. They can help to change the shape of the cell, particularly if a cell is moving, and they can support structures like microvilli. The second type of filament is the intermediate filament. Intermediate filaments are stable rods about 7 to 11 nanometers in diameter. The composition of intermediate filament varies. One of the most common intermediate filaments has keratin proteins. These types of intermediate filaments are found in the skin and are associated with nails. They are the most durable of all filaments, and their function is to strengthen cells and keep their shape. They can also stabilize the position of organelles and the cell itself. The third type of cytoskeletal structure are microtubules. Microtubules are hollow tubes about 25 nanometers in diameter, and they are composed of tubulin proteins. These are the largest of the filaments and have many functions. They are the primary components of the cytoskeleton. They can help change the shape of the cell. They can help shuttle vesicles and organelles around the cells through tiny molecular motors that walk along microtubules carrying organelles on their backs. They aid in cell division by moving chromosomes, and lastly, they form the structural components of some of our organelles. Another type of non-membranous organelle is the centriole. All cells undergoing cell division will have centrioles. We first talked about these earlier in this chapter. There are some cells that do not contain centrioles and are unable to divide. These cells include red blood cells, skeletal and cardiac muscle cells, and neurons. The structure of a centriole is composed of short microtubules. The microtubules form 9 groups, each with 3 tubules. They form the spindle apparatus that is associated with the movement of chromosomes. Cilia are structures found on cells lining the respiratory and reproductive tracts. Their structure is a long extension of the plasma membrane and are supported by microtubules that extend into the cilia. Their function is to move fluids or secretions across the surface. They create synchronized waves and can move materials very effectively. Ribosomes consist of ribosomal RNA and protein. They are about 60% ribosomal RNA and 40% protein. They consist of one large subunit and one small subunit. Both subunits must come together for ribosomes to function properly. Ribosomes can be found attached to the rough endoplasmic reticulum in the cytosol, and they are involved in protein synthesis.